Well, thanks very much to the organisers of this meeting. Oh, nearly fell off the stage. Great to be back live and to see so many faces again. And thanks really to Flav and Ho for giving me the opportunity today to tell you a bit about some of the work that we've been involved with in terms of understanding genetic modifiers at the Huntington's disease. And particularly what I'm going to tell you today is some of the work that we've been doing more recently in terms of looking at cis-acting modifiers and the potential intersection between those cis-acting modifiers and the somatic expansion that, as Jill alluded to earlier on, is now primarily from the genetic modifiers work established as a clear, important part of the pathogenic pathway in this disorder. So again, a slide that we've seen many times already, illustrating the important relationship between the number of CAG repeats inherited and the relative severity of the disease, as indicated here by the age of onset. So it's quite now well established that each single CAG repeat inherited from one generation to the next precipitates a 3-year decrease in the age of onset. And the number of CAG repeats accounts for around about 50% of the variance in observed age of onset. So clearly the most important modifier. But nonetheless, as illustrated here, for an individual with 42 repeats, there's a huge amount of variation that we see between individuals in terms of the exact age of onset that anyone will have. And that varies over probably 30, 40, 50 years in terms of the potential differences. So some individuals get the disease much earlier, and some individuals get the disease much later than we would have otherwise predicted based purely on their CAG repeat alone. And I think this is a really important observation, because trying to understand what the cause of that variance is, we think is likely to offer new windows into therapeutic opportunities. If we could understand why a subset of individuals are highly protected and get the disease much later, and we could try and apply that knowledge to these other individuals who get the disease earlier. Or conversely, if we understood why these guys get it earlier, we could try and stop whatever it is that's doing that. And it's quite clear from a lot of the data that's been accumulated over the last few years that some of the trans-acting modifiers of somatic expansion are at least contributing towards this. So many of the modifiers that have come out of the genome-wide association studies are indeed DNA repair genes that act in trans to modify the rate of somatic expansion. But still overall, we're not accounting for quite a bit of this variation. So some of that is probably going to be noise in the measurements. Some of that may be environmental factors, and again, I think that we should probably have more efforts in trying to understand what those environmental factors are, but some of it is still genetic, so there's still some additional genetics there that we haven't uncovered yet. So again, as kind of Jill alluded to, our traditional understanding of the disease is that we start with a CAG repeat that is transcribed into the mRNA, that then is translated into a protein, and it's really the polyglutamine within the protein that is the pathogenic element. And it's the increase in CAG leads to an increase in glutamines that then leads to the disease. Obviously, our understanding of that pathway is now further complicated by the fact that it's not just the inherited CAG, but the rate at which it expands is also another important driver. But still, there's this general assumption it's the polyglutamine downstream of the CAG repeat in the DNA that is really ultimately killing the cells. Although, as Jill alluded to, maybe there is potentially also a potential role for the mRNA. What I'm going to illustrate today is that, again, that we shouldn't just be thinking about the CAG, and it's not just the glutamine, that there might be other components to the HTT repeat and gene that may also be important. So again, this is data that we've already revealed. A number of groups showed a few years ago that it wasn't just as simple as the number of glutamine residues that were encoded in the protein. So the typical version of the Huntington gene has a CAG repeat that obviously codes for glutamine, but it's preceded at the end by a CAA codon and another CAG repeat that also code for glutamine. So in the typical structure, which in European ancestry populations account for about 97% of expanded HD alleles, the length of the glutamine tract is 2 amino acids longer than the length of the CAG tract. However, there are a subset of relatively rare individuals who carry variants of that, One where there is essentially 2 copies of that final cassette, so there are 2 CAAs and 2 extra CAG. So in that case, the number of glutamines is 4 more than the number of CAG repeats. And then there's another set of individuals who don't have any CAA interruptions at all, and in that case, the number of CAG repeats is exactly the same as the number of glutamines. So essentially, this provides a natural experiment in human populations to try and figure out what is the most important factor. Is it the number of glutamines that are encoded, as you might simply think, in terms of we think about what's the downstream pathogenic element that actually kills cells, assuming that's the protein, then it might be the number of glutamines that's most important. Or is it, in fact, the number of CAG repeats? And again, this work, really has been facilitated by the genetic modifiers of Huntington's disease working group that Leslie was a founder member of. So it's really, again, really fitting, I think, today that we're able to present some of the work that Leslie was intimately involved with over her career. And again, Leslie was one of those people who recognized very early the potential role of genetics to provide new insights into the disease, and I think as importantly also recognized that we were only really going to be able to achieve those aims by lots of groups working together. So I think the Genetic Modifiers group is a really fitting tribute to Leslie's contribution, and the results that come out of that will be a lasting legacy to her efforts. So thanks again from me in particular to the contribution that Leslie has made to allowing us to be involved in that aspect of the work. So a lot of these data I'm going to present you today are some of the latest results that we've started to generate from looking at the latest cohort. So the data I'm presenting now is around about 4,000 individuals from the GWAS-6 cohort of a subset of the enroll patients. And all of those patients have been we've genotyped the HTT repeat by a high-throughput MiSeq sequencing assay, so we can determine the exact number of repeats and the exact structure of the sequence as well. And again, that's, really facilitated by the amazing Enroll HD cohort of patients and the amazing data that's associated with them. So again, this just shows the classic age of onset analysis, and these are individuals with the typical allele, so they have the CAG repeat, 1 CAA, and 1 CAG as illustrated over here. And this is now based on the number of glutamines encoded. So we're just looking at the total number of glutamines now. So what about those people with the duplication? You see them that they're non-randomly distributed across this distribution. So you'll see on average they have larger alleles, and the majority of them are above the line. So if we base it on purely their number of glutamines, they have a later onset than we would have predicted based purely on the number of glutamines. Whereas if we look at these individuals over here now who have lost the CAA, they have no CAA, so their CAG equals the number of glutamines, then you can see again that they are non-randomly distributed. They are present with individuals with much smaller alleles, particularly in this low penetrance range. They are massively overrepresented in the low penetrance range, and they are nearly all below the line. So they get the symptoms much earlier than we would have predicted based on the number of glutamine residues that their alleles encode. And again, those are substantial effects. So again, probably about a 6 or 7 year delayed age of onset based on glutamines for the duplication of the CAA, and about a 12-year earlier onset for loss of the CAA based on the number of glutamines. So if we correct that for the number of pure CAG repeats, then those points now lie much closer to the line. So we're bringing everyone in now. These individuals are still probably a little bit above the line with the duplication, and these individuals here still, with the loss of the CAA, still look like on average they get the symptoms a little bit earlier. And again, the maths kind of support that. It's not quite significant for the duplication, although in a larger cohort of patients, when we combine several cohorts of patients together, that result does become significant. And with other potentially more sensitive longitudinal measures of disease severity, again, that becomes a positive association. And likewise, again, the loss is still, even when we correct for the number of pure CAGs, those individuals still get the symptoms earlier than we would have expected. So one of the questions is obviously maybe that those sequences are having an effect on the relative expansion, somatic expansion of the repeat. And we've presented data previously on a relatively small cohort of individuals in terms of how the number of repeats changes throughout the lifetime of an individual. And it's dependent on the number of repeats inherited and the age. So this is from the MySeq assay, sequencing the repeat, how much somatic mosaicism we see in blood. So an individual with 38 repeats, we see relatively low level of instability, but it definitely increases with age. And as the repeat number increases, we see more instability in this cross-sectional analysis, and there's clearly an interaction between the number of repeats inherited and the age of the individual as to how much somatic expansion we see at any one time. And that becomes quite dramatic as the alleles increase from the high 30s up into the high 40s and up to 50 repeats or more. So this is some nice cross-sectional data here. Mark Ciosi in the group has done a nice study on some longitudinal analysis where we've actually followed the same individual over 2 different time points, and he has a very nice poster, D1, and I would encourage you to go and have a look at that and talk to Mark about his poster there. So we have now huge amounts of data, over 4,000 individuals now, in terms of the amount of somatic mosaicism and how that associates with age and CAG length. So when we correct for age and CAG, we end up with a residual score here, which we call the somatic expansion score, and that's essentially normally distributed around 0. So the simple way to think about this is that all the people with a higher score than average are expanding their repeat more rapidly than average, and those who are slower expanders are expanding their repeat more slowly than average. And that phenotype is associated with residual variation in age of onset. So this is age of onset corrected for the number of pure CAG repeats inherited. And what we see here is an inverse association. So those individuals who are expanding their repeat more rapidly than average get the symptoms earlier than we would expect. Those individuals who are expanding their repeat more slowly get the symptoms later than we would expect. That correlation, although highly significant, is nonetheless extremely modest, and that's almost certainly because we don't think that it's the somatic expansion in blood that is really having any impact on the phenotype, but this is really mirroring a similar process in the brain. Obviously, we're just not able to measure directly what's happening in the brain in large numbers of individuals. And we know that some of the genetic modifiers that are modifying the rate of expansion in brain, we predict are also in blood are also modifying the rate of expansion in brain, but they don't all work in the same direction. So we've already got some data that the FAN1 variant that we'll hear a bit more about later on, it modifies the instability in blood in a different direction to the effects that we see in the brain. So that explains at least part of the disconnect there. So an obvious question then is, well, what best predicts somatic expansion? Is that the number of CAG repeats or the number of glutamines? And does that then provide any insight into how we might resolve this CAG versus glutamine dichotomy? So if we predict the somatic expansion score based on the number of glutamines, then those individuals with a pure tract have a much faster rate of expansion than we would expect, and those with multiple interruptions have a lower rate of expansion than we would expect. So this is telling us that it's really probably the number of CAGs that is better predicting that, and that's shown over here. So then if we now calculate the somatic expansion score based on the number of pure CAGs, then those two differences come back into line. And now there's no strong difference between the presence or absence of the duplication in terms of somatic expansion, although it looks like there's a little bit less. And again, in larger cohorts, that does come down to be just about significant. But actually already, the amount of somatic expansion that we see with the loss is, at least in blood DNA, convincingly lower than we would otherwise have expected. And again, this is going in the opposite direction of what we might otherwise have predicted. So again, these individuals have an earlier age of onset than we would have expected, yet they're showing slightly lower levels of somatic expansion in blood. So maybe that effect is unique to blood, maybe they do show faster somatic expansion in the brain, or maybe something else more complicated is going on. So, change track a little bit now, and again, just talk about another route into genetic modifiers. Obviously, we've made a lot of progress over the last few years in studying particularly the enroll cohort, which, as I'm sure you're aware, is largely comprised of individuals of European ancestry. That's been massively informative, but obviously it's limited in terms of the worldwide genetic diversity that's encapsulated within the enrolled cohort. So actually this is a collaboration that sprung out of a previous EHDN meeting in a coffee break after a previous presentation. So again, just illustrating the value of not just the EHDN meetings, but the face-to-face interactions that happen in the breaks and dinners and drinks at the bar, etc. So this is a collaboration with Amanda Kraus, Fiona Bain, and Jessica Dawson in the University of Witwatersrand in South Africa. So HD is rare in South Africa, and in the European ancestry population has a similar incidence as it does in Europe, around about 1 in 10,000. But it's also present in the African ancestry population there as well, but at an even lower frequency of around about 1 in 100,000. So obviously then the number of individuals who are available for research is obviously relatively limited. So we were able to nonetheless, Jessica, and with a lot of help from Mark in the lab, Jessica was able to sequence the HTT repeat from 68 individuals of African ancestry in this population, unrelated individuals. And so again, we kind of did that with the expectation that we might find some additional polyglutamine CAA type variants that might shed more light on that process. And indeed, we did detect a subset of alleles with either the duplication or the loss, but they were relatively rare. And again, remembering, although they were more frequent than we see in Europe, they were still relatively rare, given that we've only got a very tiny population. So we didn't really have enough individuals here to ask anything about that. But as Elena already told us, downstream of the CAG repeat, there's also this polyproline domain that is primarily encoded by CCG codons, but also includes some CCA codons and some CCT codons. And in the majority of typical alleles that we see in the European population, there's a single CCG, a CCA, then typically either 7 or 10 CCGs, then a couple of CCTs, all of which encode proline. But what we found in the South African population, on the disease-associated allele, there was this other very common variant that lacked the CCA proline, and had 9 CCGs. So again, the coding potential here is actually exactly the same as this allele up here. So there are exactly the same number of prolines encoded by both of these. So this is a synonymous variant we would predict has no impact on disease severity. But this allele is incredibly common in this population, so it allowed us to ask, again, independent of the number of CAG repeats, does the number or does the sequence of the proline stretch make any difference? And indeed, it turns out that we got a very strong association between the number of CCA variants and the age of diagnosis of the disease. And those individuals with the CCA variant had a much earlier age of onset than we expected. And again, that's illustrated there. And again, when we looked at their somatic expansion, again, no obvious difference between the somatic expansion dependent on the number of CCA variants. So those alleles also exist in the European HD population, again, much rarer, but again, with the beauty of having a very large population, we can also address that there. And again, we found that, again, that the CCA loss was associated with an earlier age of onset in this European ancestry population. Again, unfortunately, not quite as simple as that, because it's quite clear that there's linkage disequilibrium between the number of CAA variants and the number of CCA variants. And actually, we have now essentially 5 relatively common flavors now of the repeat, where we either have duplication and presence of the CCA, loss of the CAA alone, loss of the CCA alone, or the double loss. And now we can obviously separate this out and ask what's going on here. Duplication, again, tends to give a slightly later age of onset. The double loss clearly associated with a much earlier age of onset. Whereas loss of the single one, again, looks like it's slightly earlier. And actually, loss of the CAA alone shows no obvious difference. So again, looks like there's a difference now between loss of the CCA versus loss of the CAA. And the primary effect here, after correcting for the number of CAGs, is actually the number of CCA codons, again, which are synonymous, don't affect the sequence of the protein that's produced. Again, that's illustrated there in terms of those effects. So what about somatic expansion? Again, is that mediating any differences in somatic expansion? So again, what we see is actually that all of these structures are associated with lower somatic expansion. That might be what we expect for this allele, that is associated with a slightly delayed age of onset, but is not what we would predict for these alleles here. So again, strongly suggesting that whatever the mechanism is that's mediating this effect, maybe to do with the proline stretch, Elena has illustrated earlier, is really important in the protein. But again, remembering that these variants are are synonymous variants that don't affect the sequence of the protein that's translated. So what then could be driving that? So just some conclusions then. So age of onset clearly is driven primarily by CAG, but is also affected by the number of CAAs and also the number of CCAs. There's obviously more genetic variation out there that we can exploit in terms of looking at different populations. And again, there's an unknown mechanism for this CCA variant. So again, we need to be now thinking not just about the CAG, but the CAA, but also about the composition of that downstream proline region and how that then affects the protein in terms of not just the polyglutamine, but the polyproline, also potential effects on mRNA, and how that ultimately impacts on disease. And again, lots of potential things that could be going on here. So maybe, although we don't see an effect on somatic instability in blood, maybe there is an effect in the brain. Maybe it's to do with transcription levels. Maybe those variants are affecting the splice variant that Jill talked about. Maybe there are other proteins that bind to those different sequences in the mRNA. Maybe it's something to do with ribosome processing. So we know that proline residues in general are very difficult to translate, and maybe the exact codon sequence modifies that as well. So lots of potential mechanisms, but at the moment we don't know what it is. But nonetheless, we could argue that, again, this is potentially another insight into the genotype-phenotype correlation, that in the same way that the knowledge now of somatic expansion is suggesting new therapies, this again is another way, potentially once we understand it, may also suggest new therapeutic opportunities. And again, just finally thank everyone who's been involved with this, in particular all the patients and families who've contributed their data to the Enroll platform. Again, a totally amazing platform that's really enabled us to go to another level in human genetics in this disorder. Particular thanks to the Genetic Modifiers Consortium, and obviously in particular to Leslie's contribution to establishing that and really driving that forward. To Vilja and Mark in the group who really did most of the work that I presented today, our collaborators at the University of Witwatersrand, and CHDI colleagues, and obviously CHDI primarily for our funding. And then just finally, anybody looking for a job, come and speak to me. Thank you.
Is that working? Yes, it is. Thanks, Darren, for that wonderful talk and a great opening for the session. We have a couple of minutes now for questions. So again, we have a couple of fixed microphones. And also a couple of, there's one from Sarah.
She said it was a beautiful talk. That was it. That was it. Right, next question.
You can edit that way.
Yes. So I'll say it again, beautiful talk. So your data with blood is very interesting because as we try and develop therapeutics, for example, MSH3 inhibitors, trying to find surrogates of somatic expansion in a peripheral tissue or fluid is obviously of interest. So your data suggests that actually measuring somatic expansion rates in blood for a therapeutic readout may not be a good surrogate of what's going on in the brain, or how would you, or do you think that's too strong a conclusion?
I think that's too strong a conclusion.
Okay, that's what I wanted to ask you.
What the data suggests is that what is going on in the blood is not exactly the same as what's going on in the brain, but it broadly parallels it. Okay. But you're right to highlight, so, and as I kind of already illustrated, one of the modifiers, FAN1, at least in terms of its effect on somatic instability in blood, is the opposite of what we predict that it's doing to somatic instability in the brain. Now, we have no direct data in humans on what FAN1 variants are actually doing in the brain, but if it's all going in the way we'd expect, then we're seeing an opposite effect. But it's important to note that that FAN1 effect, actually it's slightly more complicated than that, is that primary effect is an EQTL. So the genetic modifier in humans is an EQTL, and that variant may be mediating alternative effects on expression in different tissues and different cell types. So it's possible it's increasing expression in the brain, but decreasing expression in the blood. Could explain that. And actually, when we look at now the much rarer coding variants, then they now go in the right direction. Okay? So that, I think, in terms of the general mechanisms, they're going to be broadly the same. Obviously, potential caveat is in terms of the exact stoichiometry of different proteins, and I think it's very likely that the stoichiometry of the mismatch repair proteins and FAN1 is probably at least what's driving some of the differences from cell type somatic instability, and how much that might influence your relative effect of a drug is also of potential interest. Actually, but more directly relevant is the amount of somatic expansion we see in blood. So it's very low. We can measure it incredibly accurately, and it is a great measure and has allowed us to do lots of great stuff, but it changes very slowly. And actually, if you look at Mark's poster, he'll show you some kind of data there about how much it changes. Over a 7-year period, we can easily detect changes, but over very short time periods, as would be necessary for a clinical trial, it might be quite challenging, okay? However, the data Mark's going to show you is now relatively old. We're starting now to look at individuals with larger repeats that are moving quicker. That's kind of clearly already justified. And also, with some refinements in the technology, we can probably also, again, push the sensitivity of that assay greater. So I think it's not dead yet, but there are other things we should be exploring as well.
We have time for one more quick question to see someone at the microphone.
Is that Paul down here's got a microphone? No? Or someone else?
There's someone at
Priyanka Hera from Alnylam. So when you speak of symptom onset in PO expansion carriers, I'm assuming that you refer to motor symptoms. Do you think, or do you know of anyone working on biomarkers early on in pre-symptomatic patients with the same concept?
Sorry, I didn't completely catch the question, but I think it was, should we be looking at biomarkers in pre-symptomatic patients?
Correct.
And if that's
in that PO expansion carriers, do they show the same effect early on before they begin to manifest motor symptoms?
So the majority of individuals in this cross-sectional study are symptomatic in terms of they've reached motor onset, but probably about a third of those that I showed you there are pre-symptomatic. And at least in blood DNA, there's no obvious difference between individuals who are symptomatic and pre-symptomatic, other than the fact that obviously the symptomatic individuals are on average older for the number of CAG repeats that they've inherited. So at least in blood DNA, I don't think there's any expectation to believe that the CAG dynamics is influenced by the onset of the disease per se. Now, obviously in the brain, it's more complicated because obviously cells are dying as a dependent on the number of CAG repeats. So it's going to be slightly more complicated to work out what the effects of cell death and expansion are in terms of how you resolve those to understand what really the underlying dynamics of the repeat were.
Okay, I think
you.
we have to move on. Thank you very much, Darren. It's